For the second step, if you want to collect the information regarding the hits, that is the molecules which will interact with this PCl2, then for that we have to analyze or surf the Google. So for this purpose we will say this, we can use the keyword anti-cancer phytochemical compounds targeting BCl2. You can use the keyword so that we can get the hits list or some of the information regarding the inhibitors of this particular BCl2. Okay, so like this we will collect the list of hits. If you want to identify the drugs, you can see here already a docking analysis are carried out with the BCl2. So by analyzing this, we will get the list of hits. So we can go through these articles, and then from the articles we may get the information about the chemicals, compounds which are act as a inhibitor for this particular BCl2, or the chemical compounds which will interact with the BCl2. So like this we will analyze the articles and we will get the hit list. Okay, so if you are not having any hits or any compound list with ourselves, we can also identify the inhibitor through the databases. Like if you want to get information about the plants, you can use these databases, Duke's phytochemical ethnobotanical databases, or you can use Indian medicinal plant phytochemistry therapeutics databases to get the information about the hits. For example, I will go for this phytochemical databases or this Indian Medical Association databases to get the information about the chemicals list from the plant. For example, I am going to this particular database. Okay, so I will get. So we have decided the disease is a cancer, and the plant we have to decide with this particular part, and then the target is decided as a BCl2. So against the BCl2 we'll find out the list of chemicals which will interact with this thing. So for this analysis we might take any one of the plant for our studies target. So I will select this plant, might be a Anthrographis paniculata. We can select as a plant Nilavembu. In Tamil it is called as Nilavembu. So we will take this plant as a plant material for collecting the hits to find out the inhibitors for the drug discovery purpose. So here we can browse the IMPPAT, Indian Medicinal Plants Phytochemistry and Therapeutic Databases. We can search the database. So for this purpose we can provide the name of the plant, and then you will click the search button. It will give the list of compounds which are available in the databases in this particular plant. So we will select these compounds like anthrophysite, anthropanin, anthroglopholite, 14-deoxy, 11-12 dihydroxy, anthroglopholite, and neo-anthroglopholite. So this anthrographane is a repeat, already it is there. So up to this there are about 1, 2, 3, 4, 5. 5 compounds are available. So we will take this 5 compound as a hits for the present molecular docking studies. So we are decided to analyze the hits derived from the Anthrographis paniculata as a hits chemical compounds from the plant Nilavembu against the BCl2 target. So to get the information about this chemicals, you can click each chemical and we will open them in a separate tab. So you can open in a separate tab. All these 5 chemicals we are decided for the present work. So once these pages are open, you can see here there is a drug likeness prediction. You can also check this drug likeness option. You can see this is going to, according to Lipinski rule, this compound is going to be a failed one. So we will not going to utilize this particular compound for our drug discovery, that is molecular docking purpose. So we have to this, since it is failed status, it's showing like the compound is a failed condition. This, the compound name is, you can see, andrographoside. So we'll mark it as a failed condition status here. Then we will use the other compounds for the further studies. We will also check them for the study. So you can see this andrographanin, the drug likeliness, if you check, yes, it's passed. So like this we will check all the other compounds. Andrographolide is also passed. So 2 compounds are selected, and then third compound is also selected, and then the neo andrographoside, and you can also see that it is also selected. So we select 4 compounds: andrographanin and androfolide, and then 14-deoxy 11-12 dehydrofolide, neo-andrographolide. So these are the 4 compounds are selected as a hits for the present study. Okay, so this is how we can select the hits. Or if you are carrying out some GCMS analysis during your project work, you can get the informations or the compounds from these particular output of GCMS analysis, and you can also use these chemicals as a hits for the docking studies. So according to our present docking classes, we have completed the target and the disease and target selection, and second, hit list selection. Along the hit list also we have to find out the drugs which are acting against the particular plants. So that list we can also select with the help of the drug bank, or even through the literature. You can find out the drugs acting against the BCL2. So you can find out the BCL2 drugs acting against BCL2. You can also find out drugs against BCL2. So like this we can select, and we will get the list of drugs, that is the inhibitor. You can see venetoclax you can get as a drug acting against the BCL2. So here also one more same drug will be there. So we can also get other drugs will also acting as against this particular BCL2. So these information can also be obtained through the literature. So hit list is ready. So we have selected this type of compounds. 4 compounds are selected, and then we can also select the drugs which will act against this particular BCL2. There are 2 drugs actually identified. One is venetoclax and another one is the pelitoclax. So these are the 2 drugs are decided to work in this particular docking process. So the details of the drugs are derived from the research articles published in the PubMed or any other research journals. You can get the name of these particular compounds. So we have decided the hits, we have decided the target. So 2 steps of the molecular docking process is finished. Okay, for the third step,